now it's Friday, every Friday lunchtime.
Okay, welcome to the Friday Plant Breeding Circle. Appreciate all of you being here, and we have a speaker today, Vishal Saitwal. Vishal is a cotton breeder in India with Pioneer, and he's here with us for a year as a Pioneer Fellow, and then he will return to India and continue his education and doing his research as a part of our distance program. We are pleased to have him with us, and he's going to tell us something about looks like cotton in India. So, it's all yours.
Thank you for introduction. It's a nice opportunity today. I have to talk to a diverse forum for the cotton breeding aspects and the challenges we have back in India. Good afternoon, everybody. I will be giving a little bit introduction what's going on in cotton in India, then what is the cotton scenario, the research in public as well as private sectors, the key breeding challenges we are facing out, and what are the research strategies we are going to adopt for that challenges. As Dr. Smith told, I am a commercial breeder, so we love to talk about some figures. So here are some figures. In 2011-12, there was around 40 million cotton packets have been planted. All of BT cotton hybrids I am talking about, and each cotton packet is around 450 gram each. This valued around $820 million. If I look at the total seed cotton industry back in India, the cotton seed values itself is around 30 to 40%. And that's why cotton is attracting a lot of research funding from public as well as private sector in India. Nowadays, the transgenic cotton is necessity. Around all 99% area of cotton in India is BT cotton hybrids, and the technology providers, they are enjoying the royalty benefit. The BT cotton is now a part of cotton genome, so what I feel that in future the strong genetics with native traits as well as some value added traits like weedicide resistance, fiber qualities and mechanized baking will lead the research as well as market in future. As I said, only the private seed sector contributes 99% of BT cotton hybrids, and all those are under the Gossypium hirsutum, which is a tetraploid upland cotton from US, under that background. And unfortunate to say that very few companies are focusing on a strong line development program which is necessary for a good product performance. And there is no public sector in picture here for the BT cotton hybrid so far. Transgenic cotton has impact on yield and productivity in India. Before I go to that, it has also impacted the species composition back in India. If we look in the 1990s, we have all the species grown. These two species, Arboreum and Herbaceum, they are diploids originated from India, and Gossypium hirsutum, which is a tetraploid, and Barbadense from Egyptian. All four species, they have been very well cultivated back in 1990s, but after the advent of BT cotton or transgenic cotton in this background, hirsutum background, it has declined to 4 to 5%, and the hirsutum area has grown up 90%. This has resulted into shortfall of short and extra long staple cotton fiber groups in India, because Arboreum and Herbaceum, they are known for short staple length, and Barbadense, which we know as for extra long staple and fine cotton fiber. So we are facing the shortfall of these fiber groups in India. The cotton breeding or the cotton research can be divided into two groups. Up to 2002, it was non-transgenic era, And after that, once we have first BT cotton released back in 2002, it is now a transgenic era in India. The non-transgenic era, we have released the world's first hybrid H4, and it has boosted the yields as well as area in India. But then we have seen a downfall. I'm going to discuss on that afterwards. But after the transgenic has been released, we can see the area has gone up, the production has gone up, but there is a slight increase in the productivity of cotton in India. So we have almost doubled the production because of the transgenics and increase in area, substantial increase in area. That was a good picture for us, that we have doubled the production and now we are the second largest producer of cotton after China. But if I compare the Indian cotton scenario with some of the major cotton producing countries in the world, where do we stand in terms of area? Yes, we are number one. We share around 11.7 million hectares of area of cotton back in India. But what about the production or the productivity levels? The productivity is much low as compared to the major cotton producing countries in the world. Even we are not touching to the global average of 750 here. So this gives rise to two questions. We have doubled the cotton production with the help of transgenic cottons. So can we once again double the yields to meet this global productivity level? Because we need to rise our production by around 60%. That means we have to double the production. So how do we are going to do that? Because we need a strong research infrastructure to increase the productivity in cotton. And yes, we do have the very good research structure. Since now the public sector is not there in the picture of the transgenic cotton, but yes, they are doing and they are contributing a very good cotton genotypes, and they are working on some aspects of cotton and they are contributing to the cotton research for the country. And we have a good history. The Indian Central Cotton Committee back way in 1921 was steering the cotton research, and it starts from there. And once we have formed the Indian Cotton Council of Agriculture Research, which we say ICR, and under that the All India Coordinated Cotton Improvement Project was started in 1967, which has contributed many genotypes and many good characters in cotton. And if we see, this project is spread across 21 research centers, and which covers around 15 state agriculture research universities across all the growing zones of cotton in India. These two institutes, Central Institute of Cotton Research, CICR, and CIRCOT, Central Institute for Research and Cotton Technology, gives functional guidance to this project. CICR is mainly involved in basic cotton research activities, the germplasm collection, evaluation and maintenance, whereas CIRCOT deals with the fiber quality aspects. So coming to the major contributions of public sector before I go to some other area, we have released the first hybrid in the country as well as in the world way back in 1970, which was called H4. This has ignited the hybrid breeding program, and several hybrids have been bred after this. This hybrid has given around 120% heterosis level over the existing varieties at that time and boosted the yields. After 2 years, we have released the interspecific hybrid called Varalakshmi, which was a cross between Hirsutum and Barbadans. Then public sector has contributed numerous varieties, and their varieties, namely LRA, which was a variety and a hybrid, was most adapted. It was competing well with the private sector in the non-transgenic era. Looking at the necessity of transgenic, the public sector has tried to release a variety. I'm talking about variety here, not a hybrid, because the private sector has got all the hybrids, but the public sector has tried to release one BT variety, but because of some technical difficulties, it is not well adapted in India. Nowadays, the public sector is focusing on molecular research, particularly disease and drought tolerance, and they have some transgenic events in pipeline which we expect in the coming years to be released in the country. And as I said, they are involved in bulk collection, evaluation and maintenance. Looking at the value of cotton research and the overall impact of cotton on social life of India, the government felt that they should improve not only the genotype but the overall aspect where the cotton is involved, right from research, taking the technology to the farmers, improving the marketing infrastructure of the cotton and modernization of ginning mills. So a technology mission on cotton was launched in 2002 and it has yielded very good results. There are 4 mini missions. I am not going to talk about 3 of them, but since the mini mission 1 is related to research, it focuses on genetic improvements by conventional as well as transgenic methods. Then fiber quality improvement, we are looking for male sterility sources, then cotton seed oil improvement as well as mechanization aspects. I will be talking more about male sterility and mechanization in latter part of this presentation. The introduction will not be complete if I don't talk about the transgenic cotton and what is the strength. As I said, we have released the first BT cotton hybrid in 2002-03, and now we have completed 10 years of transgenic BT cotton sowing back in India. As one can see, there are more than 1,000 of hybrids have been released in India BT events. And these are some major growing zones like north zone, central part and south part, and we have 5 to 6 events and more than 50-60 companies involved in active research of this transgenic event. But only a handful hybrids are famous, and I can share that although we have 1,000 hybrids, the genetic base is very narrow because only 2 or 3 major breeding lines have been used to develop hybrids in India. What's in pipeline? The private sector is ready with a few of next generation traits like Roundup Ready Cotton, which we say BG2 RRF. It's ready for release, waiting for regulatory approvals, which will give us the insect protection as well as herbicide tolerance. Then a white streak event from Dow, which is for insect protection. Then a spec event from JK Agri Genetics, which is from India. It's for insect protection, and a different event from Bayer for herbicide tolerance. As I said, the public sector has also initiated the research on transgenics, and they have different genes than the private ones. They are working on Cry1A, A3 and Cry1F and Cry2A. And University of Agriculture Sciences, Harvard, which is a state agriculture university, is actively involved in developing these events, and they have tested it under field trials, and they are also waiting for the regulatory approvals. CICR Nagpur has successfully transferred these 2 genes for drought tolerance. Then they are also in an initial phase of looking some genes which will improve the fiber strength, then disease resistance, particularly bacterial leaf blight and cotton leaf curl virus. The interesting thing is that public sector has in progress 2 genes under arboreum background, and these are the varieties which are known for the long staple cotton, and they have transferred the BT gene, and this may be the first record of development of transgenic in arboreum from India. Well, if I look back, I can see 2 peaks in the production. First peak after the release of hybrid, that is non-transgenic hybrids back in 1970s and 80s. After the immediate release of the hybrids, we have seen the growth in the cotton production, but then it has declined because of the prominence of bollworms. Thanks to BT cotton, because then we had suppressed the bollworm population in cotton and ultimately we have increased the yield substantially. But as a breeder, I have a question that how to maintain this level or increase this level further, because we have suppressed the bollworms, but now we are observing the resurgence of sucking pests. They are growing very fast on the cotton now, and only they can be controlled chemically because we don't have transgenic approach to deal with sucking pests. The second problem is increasing labor cost in India. This may not directly affect the production or productivity, but the farmers may go to different crops, competing crops like corn or maybe soybean, because they are more labor efficient than cotton, because cotton it's totally hand picked in India. There is no machine picking in India available right now. As a breeder, if I have to define the key breeding challenges, there are two zones. In North India, we have this viral disease, CL, which we call cotton leaf curl virus disease, and it is transmitted by white flies. This disease may cause yield losses up to 30 to 40%, and maybe if it's in high incidence, then up to 80%. So it will devast all the cotton crop there. And in the abiotic stress, we have high temperatures, waterlogging, and salinity stress in the north zone. Central and south zone, which is the major part of Indian cotton area, there we have the sucking pest. These are the plant hoppers coming, they have emerged as a major pest. Then thrips, they come eventually, and a new disease has been identified, which we say as parawilt. There is no pathogen related with this disease. 2 days before, you can see your plant very green and full of bolts, and after 2 days, if you go to the field, it will be completely dried out. So this disease is now reducing or limiting the yields of cotton in India. In total, because of this biotic and abiotic factor, we are only realizing 60 to 65% of real genetic potential of cotton hybrid. So there is much scope to improve our present genotypes, who can offer better tolerance against these biotic and abiotic stresses, so that we can realize their genetic potential. We have doubled the yields with BT cotton, and now we have to again double our yields to meet the at least the global productivity level of the world. Can we do it? Yes, we can do it, but we have to make a choice. Here we are standing, the breeders, the industry is standing at the junction, and we have to make a choice here. The present genotypes, they are all tall and robust. So the one way is to improve these tall and robust genotypes, conventional genotypes, what we call under BT background, with some of the events which are coming out in the market, and have more harvest per plant. That can be one strategy. Or to go for short, compact genotypes, which are suitable for high density and which will enable the machine picking and to tackle with the labor cost. So the second strategy will be more plants per acre, obviously. What are the traits we are looking for? What will be the traits for improving the conventional robust type in India? As I said, sucking pest tolerance is the major and priority trait for us. Male sterility, yes, we need male sterility. Drought tolerance, it was a limited focus back in India for drought tolerance, but nowadays this trait is again gaining importance. Lean yield and fiber quality, we have a less focus nowadays, but in future it will gain the importance. And disease tolerance like CLCV and bacterial blight. We can also look for these traits through transgenic breeding, and there are successful examples that people have introduced, that we have introduced Lepidoptera resistance, we are looking for sucking pest resistance, herbicide tolerance, and drought tolerance. But the thing is that we are looking at differently. Means individually we are breeding for the traits like sucking pest tolerance one group and drought tolerance second group. Can we go for multi-adversity resistance breeding in India? Because I was just going through the literature of Texas A&M, and we have a very good multiple adversity resistance program in cotton, and that has contributed well, very significantly, and improving the genotypes of cotton. And at the one time, I think, if I am correct, we have screened for 52 traits under this multiple adversity resistance program. So why don't we look for some 5-10 traits at one time to improve the robust genotypes back in India? I was talking about male sterility, so some facts and figures. The whole hybrid seed production in India is hand emasculated and hand pollinated. As I said, we produce 40 million packets, 450 gram of each, which goes up to 80,000 tons seed produced from 0.1 million acres area each year, which involves 1 million man days. And if I calculate the labor cost, it goes up to $4 million US. So this is the kind of huge task we are doing. Every single hybrid seed is produced by hand emasculation and hand pollination. If we look at the procedure, how we do that, the picture is clear. I think the picture, but you can understand. We have to find a right bud stage in the afternoon, then remove the anther stamen, keeping the stigma intact. We have to take care that this white layer of the ovary should not be damaged, otherwise it will drop down. Then cover it with the red packet. Again go next day, pollinate it and tag it. So this is the procedure we are following, and we are producing 80,000 tons of seed each year. So that's why male sterility is desperately needed for India. So the benefits, yes, male sterility can reduce the labor cost as well as the hybrid seed cost. Do we have the male sterility in India? Yes, we have male sterility in India. The male sterility comes from Hurricaceae background, which is a wide species, but the cytoplasm carries few bad genes which has got yield drags. So we have noticed that the yield potential of CMS based hybrid is pretty low than what the same conventional counterpart like A into B system and B into B system. We have also noticed that there is an issue with the stability of the restorer gene. After 3-4 years of breeding or using of the same line, we have noticed that there are sterile plants being observed in the hybrid seeds. And since there are limited number of restorers, one cannot go for a wide-scale testing of hybrids. So how we are tackling this problem? We are looking for a new male sterile cytoplasm sources, and CICR has identified that one new source from Eridon, which is again a wide species of cotton. We are also looking for genetic based male sterility, but it has got also its own disadvantages. Then we can also go back and do the intensive breeding program for the present male sterile lines as well as restorer lines to improve them further. And we may means a transgenic approach to clone these R genes, because very well identified markers are available for this R gene. So we can, if we can clone this gene and transfer it to the hirsutum, it will be a great use for the Indian farmers. The hot topic, the second option what I was talking about, that can we increase the plant density and have greater yields from the same area. Nowadays, back in India, everybody is talking enthusiastically about this approach, that is high density products. This is not a picture from India, this is from maybe from Brazil. but we hope that we can see this picture back in India after 4 or 5 years. So for these high density products, yes, the labor cost and labor shortage as well as high yield expectations are playing important role to drive this program. And currently 3 or 4 SAUs are working on breeding for suitable genotypes. Since transgenic is necessity and most of the private companies, they have these transgenic events, they are aggressively working on conversion of suitable plant type again in the Bolgat 2 conversion. And few companies are at the stage that they can release this short compact type of genotypes, I would say genotypes, in the coming 2 or 3 years. And we expect the faster adoption where the irrigated area is there and there is acute shortage of labor. Apart from working for the suitable genotype, there is a need to develop suitable machines which will enable the mechanized picking for the cotton. So the farm equipment companies are also working closely with the seed companies to develop smaller and indigenous harvesters. Few words about the product trait focus, because we have to redefine all our breeding systems. All our present genotypes, they are tall and have 2 to 3 up to 5 monopodia, which are the side branches in cotton, but here we need 0 monopodia. Then all the BT cotton hybrids currently are non-synchronous as well as they are medium to late maturity group, whereas for high density we will need synchronous and early maturity groups less than 150 days. And yes, medium height, tolerant to sucking pest and moderate bole number. So we have to shift the breeding program or breeding focus from the present to a different approach for high density product. But the basic question is whether to go for a variety for high density conditions or the hybrids. Are we going to continue with hybrids or a variety will be a better choice? If we go for hybrids, then are we going to get a better heterosis level there compared to variety? What about the seed production capability? Because at present we are producing 40 million packets, and if I go conservatively, the seed demand will be doubled, and where to produce this much of seed from the same area, that will be a big challenge for us. Seed cost, how to calculate the seed cost and whether the farmer will accept it. And the basic question is that whether the private seed companies are really interested in varieties, because they have their economic interest in the hybrids. During my PhD studies, we will be trying to find answer for the first question, that whether we are going to get a process level in the high density conditions. These are the few challenges we have to keep in mind before going for breeding of high density products. Right? We need to have suitable genotypes. We need to have suitable machines. Not only we are going to change the breeding strategy, but the agronomic practices also have to be changed for high density conditions, which is more seed rate, use of defoliants, and use of bole opening chemicals, for which we have to train the farmers. Once we shift to machine harvesting, there will be a more trash content in the cotton. So there we have to pre-clean the cotton before sending to G. So these are the few challenges we have to keep in mind before going for high density conditions. The third topic, the third option to maintain or to increase the productivity level. Most of the area in India is under rain-fed conditions, and there are marginal to low fertile soils. There, I think arboreum will be a better choice because of its many attributes. Arboreum, which is deployed from India, has got a better sucking tolerance, which is presently we are facing a problem in India. It has got a better water stress tolerance as well. Its tall and erect genotype will immediately can fit well under the high density conditions, then it has got one issue that in the late season, pink bollworm comes on it, which is the Pectinophora gossypiella, which damages it. So transgenic will be a value addition. We have now, it has got a short staple, but now we have successfully introduced a long staple from hirsutum, so we have eliminated that issue. And male sterility system is very robust and stable in Arborium as compared to hirsutum. So the seed production will be easy. And on the top, it will prevent the genetic erosion of cotton because it has got various very good attributes or gene sources in that cotton. And in fact, the Arborium genome is one of the genome in G. hirsutum if we see. I mentioned that the genetic base of cotton is narrow, so we have to go back to the white spaces, and cotton has got 50 white spaces and spread across 9 different genomes. And there are successful examples of using white spaces for transferring useful characters like acid resistance, drought, some disease resistance, fiber quality improvement, and male sterility as well. So we can go back to the white spaces, use them, and we can create the useful genetic variability. How we will be marching ahead? We will continue the improvement of conventional tall hybrids in India. Then we have started breeding for suitable genotypes for high density. At present condition, the plant population per acre in India is 7,000 to 8,000 plants per acre, and now we are trying to go up to 15,000 to 16,000 plants per acre and trying to check out how many yield gain we can get from that. And once we standardize the genotypes for high density, we can shift to ultra density types, which goes up to 1 lakh plants per acre, but it will take some time. Then, as I mentioned, the multiple adversity resistance program for key target traits will add, and we have to explore the Arborium potential, and for this we need a public-private partnership because there is a gap of sharing of germplasm between private and public breeding programs. So that gap we have to close so that to bring a good germplasm and good products to the farmer. Lastly, if we increase the productivity, how it's going to impact on the international market? Question is whether it's a direct challenge for major cotton exporting countries. I feel it's not, because the present production is 23 to 27 million tons, and the demand by 2030 is estimated around 48 million tons, which is a huge gap, and to produce 48 million tons from the existing area, that is 34 million hectares, will be a huge task. So India definitely, which has got the highest area, which is 33%, and we have a scope to improve the productivity, can play a major role to minimize this demand and supply gap. And in fact, all the major cotton growing countries have a scope to increase the productivity and minimize this gap. So with this, I invite the discussions, suggestions or questions if we have any. Thank you for listening.